Assalamualaikum viewers, welcome back. This is Dr. Abid. Viewers, from our previous lectures, we were discussing about collection of the semen to the cryopreservation of the semen. So in our previous lecture, we had a great discussion on collection of the semen, and in our today's lecture, we will be discussing about the next step of the semen collection to the cryopreservation, that is analysis of the semen quality. So in analysis of the fresh semen, the final composition of the semen depends on the level of development of accessory reproductive glands secretions and the semen or spermatozoa quality. While in the bull, these secretions, accessory secretions from the various glands discussed in our previous lecture, that includes the prostate gland, bulbourethral gland, epididymis, etc. So in the bull, these secretions are the following: 50% secretions from the alveolar glands, while 25% secretion of the semen from bulbourethral glands, 7% secretion from epididymis, 5% secretion from the prostate gland, while 14% spermatozoa produced from the testes. This is the overall composition of the semen, which consists on semen plasma and spermatozoa, as already discussed in our previous lecture also. So the chemical structure of the seminal plasma includes water 90 grams per 100 ml of the seminal fluid, protein 6.8 grams per 100 ml, fructose 460 to 600 milligrams. While this question is most frequently asked, that which one of the sugar is commonly found in the seminal fluid, that is fructose. While sorbitol 10 to 140 milligram, citric acid 620 to 800 milligram, and glycerophosphocholine 350 milligrams, while inositol 660 mg percentage. So this is the overall composition of the seminal plasma, or you may also call as a seminal fluid. However, the properties of the fresh semen of the bulls includes following: total volume of the ejaculate includes 2 to 8 ml in a bull, that is considered as a normal, and color of the semen at the time of collection should be creamy or half white, and texture should be milky or milk with creamy, and similarly fish like flavor, and spermatozoa concentration should be 0.6 to 1.5 into 10 raised power 6 in millimeter cube, while pH of the semen should be 6.2 to 6.8. So these whole parameters are considered to be normal parameters for the fresh semen of the bull. However, the evaluation of the semen consists on two parts. One is macroscopic evaluation or grace evaluation or visible evaluation of the quality of semen, which includes further two parameters, volume and color, and other one includes microscopic evaluation, which will be discussed in later in this video. While in volume, as discussed earlier, that 2 to 8 milliliter for the bull is considered normal, while the volume from the each species varies, and color should be milky white or off white in color. Here volume is greatly influenced by frequency and method of collection, the age of the male, because the quantity of the collected semen in growing bulls increases with age. Lack of preparation of the bull or mistakes committed during the collection may influence the volume of the semen, and diseases of the urogenital system greatly influences the production as well as the ejaculation of the semen from the reproductive tract. The parameters are shown here are of great importance to be considered while further evaluation or processing of the semen. Color of the milk should be milky off white is considered as a normal. However, the quality or color change in the color may be considered as abnormal or restricted to further proceedings, while it may influences or greatly losses the economically and other quality parameters for fertilization as well. For example, here shown includes pink color indicates contamination with billet, while gray or brown color semen indicates reproductive organ infection, either bacterial or viral infections, while urine contamination is usually yellow with a characteristic odor and semen appears diluted. So these parameters should be considered while evaluating the quality of the semen physically. Here, after collection, semen should be immediately maintained at the room temperature in a water bath. However, another part of the semen analysis includes microscopic examination. What is microscopic? It means that we cannot evaluate the quality of the semen through our eyes, naked eyes. It means that we need microscope to further evaluate the quality of semen. If the volume and the color of semen is normal, then we will proceed the semen for microscopic examination. Otherwise, we will reject for further proceedings to save our time and expenses. Here, if the process 1 is successful, then second part of the fresh semen analysis is a microscopic examination, in which phase contrast and a heated stretching table is used to evaluate the quality of semen, and the samples of the semen are placed on the table heated to 37 degrees centigrade and covered with a cover slip, and two types of the spermatozoa progression is examined. In the first, the progression of spermatozoan mass is analyzed, so-called weighing, and the percentage of spermatozoa of general progression as well as the level of agglutination of diluted semen. And the progression of spermatozoan mass is called weighing because it is induced by the progression of separate spermatozoa. For mass activity, a 6-degree scale is used to evaluate the quality of semen, while scale 5 includes very strong mass, that is highly recommended, or it means that congested spermatozoa quantity, and 4 includes the strong, while 3 moderate concentration of mass activity, and then 2 includes weak, 1 very weak, and 0 there's no mass indication on the microscope. Here the mass mortality is shown in the video, but I am unfortunately unable to open this video, so I will show you in the next video. The evaluation considers only those spermatozoa featuring rapid and forward progression. Other forms of the progress like circulating, backward or oscillating are assumed as incorrect. So the mass evaluation should be mass progression, or the evaluation should be unidirectional or parallel movement. The evaluation scale of the forward progression of spermatozoa is as follows. 5-degree, almost all spermatozoa are moving. It means that quality of semen is 80 to 100%, and 4 for forward progression with 60 to 80%, 3 forward progression with 40 to 60%, 2 for forward progression 20 to 40% quality, and 1 forward progression shown by few spermatozoa means 1 to 20, while 0 all the spermatozoa are still they are not forwardly progressing. Another Analytic tool that is microscopic includes the motility of the sperms, where sperms are moved and seen on the microscopic percentages evaluated. Concentration of the spermatozoon should be more than 70%. Spermatozoa observed in simians have a shape defined as a correct, particularly for a particular species. Apart from them, there are also changed spermatozoa with disqualifying defects that are called incorrect forms. And in the morphological evaluation of the ejaculate, the type and number of incorrect spermatozoa are determined in comparison to the total number of spermatozoa. Here, morphological structures are shown that includes the head, neck, and the tail of the spermatozoa to evaluate the morphological changes in the sperms. While sperm quality or morphologically sperms vary from species to species, however, the abnormalities are also shown in later slide. The evaluation also determines the presence of other cells such as leukocytes, erythrocytes, or epithelial cells. Due to the place of defect formation, two groups are distinguished. One includes primary changes formed during spermatogenesis and being the result of pathological processes in testicles, while second includes secondary changes appearing after the spermatozoon is finally formed and has left the testicles. Here, this is the diagrammatic or morphological figures shown to understand the normal and abnormal uh, figures of the normal or abnormal morphological changes in the species, like bull. Morphology of the bull sperm includes this, while this is the sheep, and this is the sperm of chicken, this is the rat sperm, and guinea pig sperm. So this is the very normal anatomy of the sperm among species, while abnormality may include the head abnormality or tail abnormality, or sometimes head and tail are detached from the neck. So these are the common abnormalities found in all type of the animals. So thank you very much for watching my videos, especially if you would like it, and hopefully you will subscribe my YouTube channel to support. Thank you very much.